Endophyte antibiotics. Does DMW 1037 and DMW 1038 show significant antibacterial/antifungal differences? Tyler and Troy Cross. The medical significance of compounds produced by different fungal species play a vital importance in the production of new antimicrobial medication that help maintain human health. Natural metabolites have been exploited by humans for thousands of years and have been the chief source of compounds used for medicine. These metabolic byproducts are the key focal point of study. By studying and testing these different products, new antibiotic compounds can be discovered and used in the fight to control the ever-growing occurrence of drug-resistant microbes. From 1983 to 1994, 60% of all approved drugs, many of which were cancer treatments, and new or pre-new drug application stages were from natural origins, as were 78% of newly approved antibacterial agents. Today, 10 to 20% of these natural origin drugs account for the most profitable of all drugs, such as penicillin. Endophytes are fungi that live symbiotically within the tissue and root systems of plants. Their metabolic byproducts are used to fight against disease-causing pathogens that try and infect the host organism, as well as other secondary metabolites that exhibit a variety of biological activities. Although the ability of fungi to produce unique bioactive metabolites is well known, endophytes have not been exploited because they are only beginning to understand what they can do. There are no new antibiotics in human trial phases currently, and that's pretty bad, pretty upsetting, because out of all the drugs or all the illnesses that we have been going on right now, we don't have any current new drugs in testing trials, so we can only use the drugs that are in stock currently. So the importance of drug discovery is paramount if we want to continue to be able to fight certain pathogens. In this experiment, we test two unknown byproducts and measure their zones of inhibition against prokaryotic species, E. coli, and a eukaryotic species, Saccharomyces cerevisiae. We compared the results and calculated the statistical differences between the two. Hypothesis, we believed that there were no statistical differences between that of DMW 1037 and DMW 1038. Our alternative hypothesis, we believe that DMW 1038 will have a stronger antibacterial/antifungal effect on pathogens than that of DMW 1037, and that there would be significant differences between prokaryotes and eukaryotes in terms of zone of inhibition. Our objectives were to isolate fungal byproducts, extract and test their properties, effect on the prokaryotic and eukaryotic species. Then we were going to determine if the byproducts could be used to manufacture novel antibiotic and antifungal drugs. We then analyzed and determined if there is any statistical significance between DMW 1037 and DMW 1038. For our methodology, we started by using sterile technique protocol to isolate and culture different strains of fungi from multiple sources. The objective of this lab was to gather a potential endophyte subject and eliminate any surface colonizing bacteria so the internal fungi could be targeted for isolation. This was done by using a combination of 0.5% bleach, 70% ethanol, and sterile distilled water rinses in between. And behind the scenes, endophyte sequencing is taking place to determine the species and source. We then use aseptic technique to inoculate pure culture species and transfer them into a growth broth. This is done with a goal to increase the fungal biomass for the extraction of fungal secondary metabolites. We did this by cutting the fungal species into small pieces and placing them in a shaker flask containing 100 milliliter liquid PDA broth. We extracted fungal secondary metabolites using a chemical solvent via solid-liquid extraction and solvent partitioning. The raw fungal extract will be tested for antimicrobial activity and for chemical profiles. We did this by blending the culture and adding ethyl acetate to create a slurry. We then used a vacuum filtration with a Brunner funnel and transferred the liquid content to a separation funnel. We collected the organic separation, which was the metabolite, and dried it in another beaker. We then performed a Kirby-Bauer test using Staphylococcus aureus, E. coli, and Saccharomyces cerevisiae as a microbial spread culture. This was done in order to test our fungal metabolite and determine whether or not it is effective against these different bacteria. In our discussion, we only addressed the prokaryotic and eukaryotic species, and those were E. coli and Saccharomyces cerevisiae. But back to this step. We did this by dissolving our fungal metabolite in DMSO to put it into a solution. Use the spread plate method to lay our bacterial lawn, placed 6 filter paper discs down on each plate, and pipetted approximately 12 microliters of our metabolite on each disc and incubated.
This graph is showing the results for our endophyte GMW1037 versus its control, and this is for E. coli. The average for our endophyte had a mean value of 9.25 millimeters for its zone inhibition, and our mean average for our control had 12.8 millimeters. Our p-value for these two samples was less than our significant level of 5%, and it was 0.0308. This means that we can reject our null hypothesis and move towards our alternative hypothesis, stating that our endophyte sample GMW10038 had more powerful potency.
Here we go, and we see the comparison between the two endophytes, not regarding the control, DMW 1037 versus DMW 1038. And this is strictly for E. coli. The comparison between these showed that the mean was of our own metabolite, which was GMW 1037, was again 9.25 millimeters, but for the comparing endophyte, which was DMW 1038, was 13 millimeters. This again showed a p-value that was less than 5, which was our significant level, and it was approximately 0.008, which was less than 1%. This allowed us to reject our null hypothesis again, and revert to our alternative hypothesis, showing that DMW 1038 had a significant difference. you can see here in the graphical representation, we have zones of inhibition on our y-axis and DMW 1037 control and DMW 1038 shown. The zone of inhibitions, as told previously, were as follows. The DMW 1037 showed a zone of inhibition of 9.25 millimeters. The control, which was significantly higher, was 12.83 millimeters, and slightly less was our comparative endophyte, which was 1038 and was 13 millimeters. The p-values, shown with asterisks, show that there's a significant difference between these endophytes and the control, meaning for us to reject our hypothesis again and revert to our alternative hypothesis. Here are the results shown for DMW 1037 and our control, S. cerevisiae. We see that the zone of inhibition that was tested showed that our endophyte had a 4.5 millimeter zone of inhibition compared to the significantly larger control, which was 21.5 millimeters. The p-value was less than 5%, and it showed that we again can reject our null hypothesis and revert to our alternative hypothesis. The p-value was 8.3 times 10 to the negative 8th. As you can see from this table and graph, these are the comparative between the two samples of S. cerevisiae from DMW 1037 and DMW 1038. The zone of inhibitions between these are slightly smaller, but the mean again for our endophyte was 4.5 millimeters, and for a comparative endophyte was 12.3 millimeters. The p-value was slightly larger compared to the control, and between these two endophytes was 3.69 times 10 to the negative 6. Again, as we have seen in previous results, our p-value was significantly less than our significant level of 5%, and we again can reject our null hypothesis. Next table, we can see the antibiotic activity of S. cerevisiae between our two comparative endophyte samples and the control. As we can see, the blue, which was our DMW 1037, is 4.5 millimeters for the zone of inhibition. The control was extremely large with 21.5 millimeters, and our DMW 1038 was 12.33 millimeters. The p-values shown on the right have an asterisk next to them showing that there is a significant difference between our comparative endophytes and the control, as well as the two endophytes by themselves. For this graph, we directly compared a prokaryote versus eukaryote, which was an E. coli versus S. cerevisiae. The average zones of inhibition of E. coli was 9.25 millimeters, and the average mean for S. cerevisiae was 4.5 millimeters. Again, these showed a p-value of less than 5%, and we could reject our null hypothesis in saying that there is a significant difference between the effects of our endophyte on prokaryotes and eukaryotes. Because we compared them directly, here's a graph representing E. coli versus S. cerevisiae of DMW 1037. We can see here that E. coli had a larger zone of inhibition, meaning our endophyte culture taken from DMW 1037 was more profound on E. coli versus the S. cerevisiae, or the eukaryotic specimen.
graph representing a direct correlation between E. coli and S. cerevisiae. But instead of our endophyte, we compare the endophyte of DMW 1038. Like our endophyte, which was DMW 1037, the antibiotic activity had a substantial influence on E. coli, with a zone of inhibition around 13 millimeters versus S. cerevisiae, which had a zone of inhibition around 12.3 millimeters. This shows that this particular endophyte had a better antibiotic effect on prokaryotes versus eukaryotes.
The goal of this experiment was to collect a leaf and extract the endophyte inhabiting the leaf to extract antibiotic metabolites that could be present. From the natural metabolites, which the endophyte creates, we could test effectiveness of both their antibacterial and antifungal properties and test their efficiency between prokaryote and eukaryote. We found that the metabolites are more effective towards prokaryotic organisms such as E. coli than fungal agents like S. cerevisiae. We suspected prokaryotes would be more susceptible to these simple endophyte metabolites because prokaryotes themselves are structurally more simple than a eukaryote. As stated by Collins, depending on the configuration of the cell membrane for that prokaryote could lead to its own demise. He gives an example of S. aureus, which lacks a D-alanine, which could make it more susceptible to antimicrobial activity. Further inspection would be needed to identify if this was occurring in our endophyte versus E. coli. Structure complexity could lead to the significant difference between the two test subjects. Why did we see significant difference between prokaryotic organisms and eukaryotic organisms in terms of their p-value in DMW 1037, but we didn't in DMW 1038? Most likely, these are a result of lack of consistent data. Our class had problems with collecting data that matched close to our control. Another reason could be the lack of results stemming from particular endophytes. Due to different endophytes, we might have compared one versus another, and they might have not have been close in origin. We would also need more trials to make sure that these are definitive results. If we were to look further into broad metabolites that our endophyte could combat, how would we go about doing this? We would need to test different microorganisms, have a broader range of testing, and we also need to test their effects on more virulent pathogens. DMW 1038 had more powerful antibiotic effects on both E. coli and S. cerevisiae, probably due to further unknown metabolites. We would need sequencing data to research more into particular species of endophytes and why it needs different metabolites to ward off bacteria or fungal agents. We will most likely need the location, the niche that it's located in, and probably competitors to verify this fact. So in conclusion, the p-values for the comparison between E. coli and the S. cerevisiae were all less than the significant level set at 5%, other than when we tested them directly using the DMW 1038. This means that we rejected our null hypothesis and look at our alternative hypothesis, which states that DMW 1038 has more antibiotic potency and does better than DMW 1037. We also found that the metabolites created from our endophyte had more antibacterial effects than antifungal effects, which can be seen when we compare S. cerevisiae and E. coli directly. Based on these results, we can't conclude that the metabolites will work for definitively for prokaryotes or for eukaryotes. We can only tell you in terms of our results, showing that it had a stronger effect on E. coli, which was a prokaryote, versus our fungal agent or Cerevisia.